The last section of this unit is on the senses. Sensory receptors are neurons that react directly to stimuli from the environment. They're part of the sensory division of the peripheral nervous system, and there's 5 different types. Pain receptors or nociceptors detect chemicals released by damaged cells. This indicates danger, injury, or disease. Pain receptors are found everywhere in the body except for the brain. So the brain is able to perceive pain in other locations, but the brain itself cannot feel pain. Thermoreceptors detect variations in temperature. Mechanoreceptors are sensitive to touch, pressure, stretching, sound, and motion. Chemoreceptors are sensitive to chemicals, and photoreceptors are sensitive to light. The first sense we'll talk about is our sense of vision. Vision is our primary sense. About 70% of all sensory receptors that we have are located in our eyes. Eye anatomy. The sclera is the white outer layer of the eye, which is continuous with the cornea. The cornea is the transparent front cover where light first enters the eye. Aqueous humor is the clear fluid filling the chamber behind the cornea. It provides the eye with nutrients as well as to help maintain eye pressure. The iris is the disc-like colored muscle that regulates the amount of light entering the eye. The pupil is the opening within the iris. It's the part of our eye that looks black. It's actually an opening that allows light to pass through to the lens which is behind it. The size of the pupil is controlled by the iris muscles to regulate the amount of light which can actually enter the eye. The pupil will constrict in bright light, and the pupil will dilate in dim light. The lens focuses light on the retina. The lens is attached to small muscles that have the ability to change the shape of the lens to focus near versus far. If you're looking at something that is a distant object, the lens will flatten. And if you're looking at something that is near, the lens will have more of a bulge to it. The vitreous humor is a transparent, colorless, gel-like fluid that fills the space between the lens and the retina, and it helps to support the shape of the eye. The retina is a thin layer of tissue which lines the back surface of the eye. It contains photoreceptors which convert light energy into nerve impulses, which are then sent to the brain. There are 2 specific kinds of photoreceptors. Rods are light sensitive, and cones are color sensitive. The optic nerve transmits visual information from the retina to the brain. The blind spot is where the optic nerve exits the back of the eye. There's no photoreceptors in this specific area, which means if light is directly focused on this location, you will not be able to see anything. Here are some images of the optic nerve. Here are some different vision conditions. Emmetropia is normal vision. This is when the image that is being looked at is focused directly on the retina. Myopia is more commonly referred to as being nearsighted. And this is when an image is focused in front of the retina. And so by the time it does reach the retina, it is once again out of focus. Myopia can be corrected with a concave lens. Hyperopia is more commonly referred to as being farsighted. This is when the image is focused behind the retina. So when the image reaches the retina, it has not yet been in focus. Hyperopia can be corrected with a convex lens. Here is a comparison between myopia and hyperopia. Astigmatism is an imperfection in the curvature of the eye, and this curvature could be on the cornea or on the lens. What this results in is that the image is projected onto multiple focal points on the retina, which means that the image that's perceived by the brain is blurry. Astigmatism often looks like this. Reading text when you have astigmatism can cause eye strain or headaches. Astigmatism is often corrected with a variable thickness lens that causes there to be just one focal point on the retina onto which the image is projected. The next few slides go over some fun facts regarding our eyes and our vision. The next sense will be hearing as well as vestibulation. The ear can be divided into three different parts: the outer ear, the middle ear, and the inner ear. The function of the outer ear is to collect sound waves. The parts of the outer ear include the auricle, which is the external part of the ear, which is composed of cartilage, and it catches sound waves and directs them into the inner structures of the ear. The auditory canal is also called the ear canal. It transports sound waves, and the tympanum is also called the eardrum, and this vibrates due to sound waves. The tympanum is the dividing part between the outer ear and the middle ear. So the middle ear transmits and amplifies the sound waves. It consists of the tympanum, which, as said before, divides the outer ear from the middle ear. And then it also contains the ossicles. The ossicles are the three tiny bones we have in each ear. We have the malleus, the incus, and the stapes. The malleus is also called the hammer. This is the bone that's attached to the eardrum. The incus, also called the anvil, is the middle bone. And the stapes, or the stirrup, is the final bone that touches the cochlea. As the sound waves vibrate the eardrum then causes the ossicles to have that vibration as well and transfer it eventually to the cochlea. Here are the three ossicles, the malleus, incus, and the stapes. These are the three tiniest bones in the human body. So then the third part of the ear is the inner ear. And the function of the inner ear is to transform sound waves into electrical impulses. The inner ear also contains the vestibular system, and this is what provides the brain with information about balance and spatial orientation. The structures of the inner ear include the oval window. This receives amplified sound wave vibrations from the stapes, or the stirrup, the last of the three ossicles. The oval window is on the cochlea, which is a spiral-shaped organ containing mechanoreceptors that transform sound wave vibrations into electrical impulses, which then can be sent to the brain. The cochlear nerve is what transmits those impulses from the cochlea to the brain. We also have a eustachian tube, and the eustachian tube has no function in hearing, but it does have the ability to drain fluid from the middle ear to the throat. And then we have the semicircular canals. The semicircular canals are the vestibular structure. They're filled with fluid, and the fluid moves as we move. There are mechanoreceptors in the semicircular canals, and as the fluid moves, it moves the mechanoreceptors, which then convert that motion into electrical impulses. So here are the different parts of the ear. Here are some animations about hearing physiology. And the next couple of slides go over fun facts about the ear and hearing. The next senses that we'll discuss are smell and taste, which are our two chemical senses. We'll start with the sense of smell. So olfaction is what we call our sense of smell. And the structures that function in olfaction are the olfactory epithelium, which is specialized tissue on the roof of the nasal cavity that consists of our olfactory neurons. The olfactory neurons are the chemoreceptors in the olfactory epithelium that detect chemicals in the air that we breathe. The olfactory neurons can detect over 1 trillion different odors. The olfactory bulb sends messages from the olfactory neurons to the brain. Gustation is our sense of taste, and the structures that function in gustation are the papillae. These are small bumps on the tongue, and these contain thermoreceptors and our taste buds. The taste buds are located in the papillae on the tongue, and they contain the gustatory neuron receptor cells. The gustatory neurons are the chemoreceptors that are found in the taste buds, and they can detect 5 different chemical types in food. They can detect sweet, which is detecting sugars. salty, which is detecting minerals, sour, which is detecting acids, bitter, which is detecting bases, and umami, which is savory, and it's technically detecting the amino acid glutamate. All areas of the tongue can detect all taste sensations. There are regions, though, that are more sensitive to particular tastes. Flavor is the sensory interpretation of food that's primarily a combination of smell and taste. Taste and flavor are different. So, taste is the detection of chemicals by chemoreceptors, which is then interpreted by the brain. Flavor is the intermingling of taste and smell receptors, and that occurs because the nasal and oral cavities converge at the pharynx. 75 to 95% of what we think is taste actually comes from the sense of smell. Remember that we detect over 1 trillion odors, but only 5 tastes. In addition to the combination of smell and taste, there are also other lesser sensory contributions to flavor, such as texture, visual appearance, and temperature. The next few slides go through some fun facts about taste and smell. The last sense is our sense of touch. And our sense of touch is determined by our skin mechanoreceptors. These are sensory cells which respond to mechanical pressure of distortion, specifically light touch, deep pressure, stretch, and vibration. These mechanoreceptors are found in all regions of the skin. We do have a different number of receptors in different areas. And the more mechanoreceptors an area has, the more sensitive that area will be. The sensory homunculus is a representation of where in the brain touch is processed for different body parts. It has a distorted appearance. The larger body parts on the homunculus mean they contain more mechanoreceptors. And the homunculus man is an exaggerated figure showing the more or less sensitive areas of the body. And then finally, the last few slides are some fun facts regarding our sense of touch.